Mr. Bobby.
Hello.
It's nice to meet you. I'm good. Please have a seat over here.
Thanks for having me over today. How are you doing?
I'm good. I don't have a lot of time, so let's just get this meeting started. You said it was something about some CRISPR, Lyme disease, and gene drive thing or something?
Exactly. Hoping you'll invest in my research and focus on preventing Lyme disease using CRISPR technology.
Alright then. Start from the beginning. So what even is CRISPR?
Alright, well that's a very good question. The CRISPR stands for Cluster Regularly Interstate Short Palindromic Responses. It's a gene editing technology that allows scientists like me to change DNA sequences and modify how genes function. It was originally discovered as part of a bacterial defense system against viruses, but scientists have adapted it as a tool for editing genes.
Okay, but how exactly does it work? Like, you gotta tell me. I'm just a business.
That's fair, right? But CRISPR mainly relies on two parts. This is guide RNA and a Cas protein, which is usually called Cas9. The guide RNA is designed to match a specific DNA sequence and leads the system to the exact target in the genome. Then the Cas9 enzyme acts like molecular scissors and cuts the DNA at that location. Once the DNA is cut, the cell naturally repairs it, and scientists can use that process to insert, remove, or disable genes, which is a type of mutation.
So what exactly does this have to do with Lyme disease?
Well, scientists are trying to use this new, profound CRISPR technology to genetically modify ticks so they no longer spread Lyme disease. Lyme disease is caused by the bacterium Borrelia burgdorferi and is mainly transmitted through black-legged ticks, which are also called deer ticks. Since ticks are the main carriers, researchers think that by stopping the disease altogether from the source will make a great impact.
And where does the gene drive thing come in?
Well, normally there's only about a 50% chance of passing the gene to offspring through standard Mendelian inheritance. However, gene drives change that by increasing the inheritance rate to over 90% and allowing a modified gene to spread very rapidly through an established population.
You know what? That sounds kind of insane.
So, it is indeed pretty powerful. But scientists ran into a major problem early on. For CRISPR to work effectively, the editing material has to be introduced into developing embryos. Usually tick eggs are coated in a thick wax layer that keeps shattering the tiny glass needles that are used for CRISPR injections.
So basically what you're telling me is that you're wasting my time with a problem that hasn't been solved yet, right? Is that what you're telling me?
Well, Mr. Wang, I would never waste your time. I was actually going to say that researchers developed another method called remote control, which means instead of injecting the eggs directly, scientists remove the organs to produce the wax coating before the eggs are laid. Then they would use a small peptide molecule that targets receptors on the mother tick's ovaries, and since the ovaries contain the developing eggs, the CRISPR components can still reach future offspring without breaking through the wax layer.
So what exactly are the researchers testing?
So they're testing whether the edited ticks can become resistant to the Lyme disease bacteria or lose the ability to transmit it. They're experimenting with genes connected to immunity, reproduction, and transmission to see whether future generations of ticks can stop carrying the disease entirely.
And how are they testing all of this?
Well, first, scientists identify a target gene and create guide RNA that matches that DNA sequence. Then the CRISPR-Cas9 system is delivered into the tick ovaries using the remote control method. So, using the remote control method, the scientists identify a target gene and create guide RNA that matches that DNA sequence. Then the CRISPR-Cas9 system is delivered into the tick ovaries using the remote control method. After editing the genes, the researchers observe whether the modified ticks can still carry or spread the bacteria. and whether the altered genes are inherited by future generations or not.
So has this actually worked out yet? Like, how can you tell me that my investment is going to be worthwhile? Because I want to make this money, right?
So far, scientists have successfully edited tick genes in laboratory experiments, which is considered nearly impossible before. Right now, though, the research is still mostly in the preclinical stage, meaning it's being tested in labs and controlled environments rather than in real life.
So what you're saying is that there isn't an actual product out yet. Why are you wasting my damn time, you filthy scoundrel? You're all the same! What do you have to say to that?
Well, there's nothing on the market right now for Lyme disease prevention. But there's currently no approved CRISPR-based product on the market for this specific use. However, CRISPR itself has already been approved for certain human diseases, like sickle cell disease, which affects the red blood cells, which shows that the technology can work successfully in the future.
So, what is the benefits of all of what you just said? What is the benefit to society? Maybe to me even?
Well, Lyme disease is the most common vector-borne disease in the United States, especially in the Northeast. And cases only keep increasing every year. Current prevention methods only rely on removing ticks quickly or avoiding tick bites altogether, but those methods aren't always reliable because they don't focus on the outcome. If scientists can reduce the ability of ticks to spread Lyme disease in the first place, it can prevent thousands of infections before they happen.
I guess that sounds useful, but surely there's a catch, like nothing is perfect.
Well, you do in fact bring up a very good point, and there are definitely many concerns. One major issue is that gene drives spread very quickly into a permanently altered ecosystem. If scientists accidentally affect other species or disrupt food chains, there could be many unintended consequences. There are also some ethical concerns about humans genetically modifying wild populations because once the gene drive spreads, it can be extremely difficult to reverse.
So if I were to invest in your product, what you're telling me is that I'm gonna get flamed, potentially canceled, potentially disliked by the entire world? You know what? That sounds like a good deal to me. I like that. Count me in.
Exactly. That's why scientists are being very cautious and continuing to study the long-term environmental effects before considering any large-scale release.
You know, you really are a good talker. Perhaps it would interest you if I scheduled another meeting with you sometime.
So, does that mean you'll invest in me?
Let's just say I am interested in learning more about your topic, perhaps about you, and what your passion is about this oddly strange. As an investor, I don't know why you chose this topic. I could never, do those science things, but I'm really looking forward to this next meeting.
Well, thank you for having me over. See you then.
I'll see you then. It was nice meeting you. Thank you. Let's go. This way. Push the chair. Yes. Okay. Let's go. So what compelled you? I'm very, very interested in your answer. What compelled you to research this specific topic? Like, you could have done any other topic, but you chose this one. Why is that?
Well, when I was 6, maybe 7 years old, my best friend got Lyme disease from a tick when we were out in the middle of the woods. There's some bond. I can't remember the name, and then I remember very clearly how he reacted in his final moments. Let's just say it's not very pleasant.
Yeah, Joe, it was a great day hiking out here. It's been a really nice day. Joe. Joe, what's wrong? you good, Joe? Joe. Joe.
Oh, I'm sorry to hear that. So I guess let's change the topic. What would you say is the most interesting thing about this topic, this CRISPR gene editing, gene drive, tick thing?
Oh, sorry. The story of my friend really made me tear up. Honestly, I think the most interesting part is that scientists are using a natural bacterial defense system and turning it into a tool powerful enough to potentially stop diseases before they spread. The idea that changing a few genes in ticks could potentially reduce Lyme disease cases across the entire population is pretty incredible. What's also interesting is how researchers had to overcome unexpected problems, like the wax-coated eggs, by developing completely new techniques like rebound control just to make gene editing possible.
You know what? I like your rhetoric. I'm definitely going to consider investing into this. What was the retainer again?
Thank you, sir. Thank you. It really means a lot to me. Based on what my team and I calculated, it's around $50,000.
Alrighty, I'll wire the money to you by Friday, I guess. Friday sounds good, right? 2 days maybe?
Sounds great.
Okay. I'll make sure the money's in there. You make sure that you get your research nicely done. Make sure you don't spend it on anything else. Drugs, something else, right? I am expecting good returns, okay? I'm looking at you. Hey, don't look over there. Look at me. I'm expecting good returns, okay? Thank you.
So, our opinion on the use of this technology is that it will be highly impactful to the society that we live in, despite the possible concerns that may come with it.
It can help other people not have to experience the grief, such pure, despicable grief that Aiden felt after his friend died because of Lyme disease from a random tick. Even if Lyme disease is rarely fatal, any possibility that one can die should be ruled out impossible.
Even if the person that is affected is Daniel De Los Santos.
Thank you.